Okay, everyone, welcome to this week's lecture on chemistry. We're going to be talking about atomic identification and imaging. Mainly, we're going to be talking about spectroscopy, absorption, and we're going to talk about IR spectra, mass spectroscopy. We're going to be talking about NMR, both proton and carbon. And yeah, so let's get started with that. Here's my information for those who are just tuning in, along with my email, which you are free to contact me through if you have any questions. We also have a GroupMe page where you can join and we can basically talk with any of the tutors, or there's a lot of students there, so you might find some helpful information from us or from some of the other students. Okay, so learning objective. What is the absorption spectrum? How does molecular structure affect absorption? Like I said, we're going to be talking a lot about things like IR spectroscopy, which does use absorption as its main principle. Then we're also going to be talking about NMR and mass spectroscopy. Okay, on to the content. First, we're going to talk about the absorption spectrum. So as we covered previously in our physics lectures, waves have varying wavelengths, which can change their properties. So light waves have come in a spectrum of wavelengths and as a result frequencies, and this determines what types of light waves they are, whether they're visible light, UV light, infrared microwaves, anything like that. Okay. For example, there's a certain wavelength range that allows us to see the light waves, and that's called the visible light range. That's what you're used to. And within this range, different wavelengths produce different colors. Red light is a different wavelength than blue light, which is different than green light, which is different than purple light, etc. When this light hits an object, part of the light is absorbed by the object and the rest is emitted from the object. What we see on the object is emitted light. So for example, if I was to, so you can see that my shirt appears to be blue. What happens is when the white light, which has all of the wavelengths, hits my shirt, the material in it absorbs all of the wavelengths of light, the red, the green, the purple, the orange, the yellow, and the only thing that it doesn't absorb is the blue light. So it reflects the blue light back. And when you look at it, you're seeing the blue light that's being emitted back, right? That's not being absorbed by the material. So it appears blue because that's the only color, the only wavelength of light that's not being absorbed by the object, okay? This happens, of course, in biology as well. And one common example of this that some of you may have seen in a bio lab is chlorophyll. You guys know that chlorophyll in plant cells is usually green. It has a green color. And here's a graph that shows the absorbance of different types of chlorophylls and carotenoids at of different wavelengths of light. So you can see that most of the time chlorophylls, they absorb purple, blue, red, and orange light very well, as you can see here, but they have very, very low absorbance of like this green, yellow light, right? So when you look at these, chlorophylls or anything through a microscope or something like that, you'll see that they appear green because that's the color of light that they are not absorbing. So they have a green color or green appearance. So that's all good, and we've learned most of this before. So what does that mean for today's lecture? So the idea of absorption can and is used a lot in experimentation to visualize changes. One common use in a biology chemistry experiment is to use absorption to track the progress of a reaction. This is a reaction that a lot of you know, photosynthesis, and some of you may have even done this experiment with photosynthesis where you can actually track the rate of photosynthesis in a plant cell. And you can change the factors. So for example, you can see what's the rate of photosynthesis when I put it in outside in the sunlight, what's the rate of photosynthesis when I put it next to like a heat lamp and keep it there, and what's the rate of photosynthesis if I keep it in complete darkness. And you want to see the difference of how like light affects photosynthesis. Well, how you would measure the rate that the reaction is proceeding is through this, at least one of the ways is through absorption. For example, if you put chlorophylls into different tubes and you add an indicator, which in this case is DPIP, but you don't really have to know that. This indicator, it turns from a blue color to a clear color during the process of photosynthesis. The exact way it does this, you don't have to know, but it acts as a substitute for one of the intermediates, and it can accept one of the electrons and get reduced instead. And when it gets reduced, it changes from blue to clear, but you don't have to know that. And so when it does this, you can measure the rate of photosynthesis by simply finding the absorption blue light over time. So you would put the chlorophylls in a test tube, you would put the indicator in, it would spin around and it makes it blue. Then you would keep it in, say, keep it outside in the sunlight, and then every, say, minute, you would measure what's the absorbance, every minute, and then you can record it, and you can just make a graph of it over time. You can plot it on a graph, and you can find it, and you'll have a slope. You'll have different time points. You'll have a slope, and that slope will be your rate, and that's the rate that photosynthesis is occurring in this specific cell. And so when you have something like this, you can actually like see the way it goes from blue to clear. And the way you do that is through a specific tool. You put the test tube in, like I mentioned, you have a test tube, goes from blue to clear. Every minute you take the test tube and you put it in this machine. You set the machine to specific wavelength. So you want to set it to the wavelength for a blue light. It would shine a light through the test tube, and when it goes through, it'll give you like a percentage or like an amount absorbed, and it'll be some number, whatever. And if it's blue, so at the start you expect it to have a very low absorbance because it's absorbing most. It is blue, so that means it's not absorbing blue. Does that make sense? It absorbs all the other colors except blue, so it actually is not blue. And then as it gets more and more clear, it'll be absorbing more and more of the blue light into itself, and that's why it won't be appearing blue anymore, right? And that's just kind of like an example of an experiment. You don't really have to know like the specific experiment or anything like that. It's just maybe one thing that you've done before. Okay, let's talk about the mechanism for this absorption. And the way the absorption is measured is based on electronic orbitals, which we've covered a little bit before, but here is an example of the orbitals. So you have the sigma bonds, the pi bonds. Remember, sigma bonds are like the first bond, and then the pi bonding is the double and triple bonds that's shared. This is the non-bonding, which is where the lone pairs, right? And the anti-bonding, which actually, as it says here, these are normally empty, but these are like to kind of prevent. These are kind of like, as you would think from anti-bonding, these just remove the bonding essentially. Okay, but again, it's not super important. Just remember that the energy levels are important. So it starts off sigma bond, then pi, and then non-bonding, and then anti-bonding, right? I remember it costs more energy to break a sigma bond than does pi bond, so sigma is higher than pi. It's easier to form a sigma bond than a pi bond, so sigma is below pi in terms of energy, right? And the reason that you want to go from non-bonding to bonding, normally from lone pairs to bonding, is because it's lower energy, right? Remember that all light has energy in it based on the formula E equals hv, where h is Planck's constant and v is the frequency of the light, or E equals hc over lambda, which is kind of, I think, in some ways easier to use. Depends what they give you, whether they give you the frequency or they give you the wavelength, because you already know what speed of light is, where c is the speed of light and lambda is the wavelength. So you can find the energy that all light waves have. And then when this light, which has energy, hits a molecule, it transfers some of the energy to the electrons. The electrons get excited and use that energy to rise to higher energy orbitals, right? So it can go from, as you can see here, from sigma bonding to a pi anti-bonding. It can go from sigma bonding to sigma anti-bonding, right? It can move up. The electrons from lower level orbitals get the energy to move up to higher level orbitals, and that's called excitation because they're higher, they're excited. Okay, the exact amount of the certain wavelength of light absorbed is dependent on what orbitals contain electrons and how much energy they need for excitation, right? Different configurations, bonds require different wavelengths and therefore different energies of light to be excited, and so that would be the wavelength of the highest absorption. I know that was a little bit confusing, but essentially what it's saying is based on how the electrons are assembled for each different molecule in these sigma bonding, pi bonding, and non-bonding orbitals is what plays a role in what wavelength would be best for absorption. Because remember, wavelength is directly tied to energy. So the shorter the wavelength, the higher the energy. If it needs to go up a lot to be, if, for example, the molecule needs a lot of energy to be excited, it would absorb very well a short wavelength, right? It would like to absorb a short wavelength, or might want to absorb a longer wavelength and go to a different level, or something like that, right? It's based on what is more favorable based on like the exact amount of energy it needs for exact excitation, right? And that's based on its orbital structure, right? And each molecule usually will have like fairly different orbital structures, right? Okay, so that's the basics of just absorption, right? Next, we want to move on to spectroscopy, right? And the first one we're going to look at is actually one that's extremely tied closely to absorption, which we just talked about. It's called IR spectroscopy, and the IR stands for infrared, right? Which remember is a certain wavelength of light. Infrared, it's slightly lower energy and higher wavelength than visible light. Okay, so this is one very common type of spectroscopy that many of you have likely seen before. The main principle is based on the absorption we just covered, specifically for the IR region. You can also do UV visible light spectrums, but those I think are a little bit less common, and those are not used necessarily for identification in this way. They're more used to identify whole molecules that have known UV visible light spectrums, okay? But this is something that they would ask you more on like an MCAT, this type of spectroscopy, and this is one that you probably learned in organic chemistry class. So you can see here, you have the infrared spectrum for hexanol. It has this transmittance, it has this wave number, which is not wavelength, but wave number. Transmittance is basically how much it passes through, essentially. And you have this little region, which is not really super important. This is like the base zone, I guess, and you don't really pay attention to this region almost ever. It's really starting from like, I think here and left that you are really looking at. This region is kind of like the background noise, I guess, kind of. It'll be there like for most of the spectrums that you see. And what you're looking for is these peaks, right? This one, and this one. And really what you're looking at is where on the x-axis they are. So where does this correspond to? This appears to be about 3,300. This is about 3,500. This is about 2,800, right? Okay. And you're also looking at the size. This is a little bit of a thicker, wider peak, whereas this is a very short peak. This is a somewhat short peak, but very intense, as in it goes down a lot on the y-axis. So just keep these descriptions in mind. And this topic is very much, you have to just memorize it, and that's about it. Just memorize and apply. There's not much thinking about it. And I will show you here, this is what you have to memorize. This table is very important. This will basically describe what the peaks for every single type of bond that you will see look like. So a benzene ring is 600, will have a wave number about 1,600 and 1,500. C double bond N will have 2,200. C double bond C, it's basically the same thing, 2,200. C triple bond C, sorry. C double bond C will be less, about 1,650. Same thing with C double bond N. C double bond O will be about 1750s, but it'll have a very strong peak, like you can see here. It's a very intense peak, right? Goes all the way down essentially. Whereas these are more mediums, these will go like halfway down. So pay attention to this. This one, OH alcohol, this one's pretty important. Broad. These guys, the OHs, they're very broad, as in they're very wide. Okay, you can see that's what it is here. Okay. And again, this is the cutoff for what it's actually important. This is kind of like the background noise you're not really paying too much attention to. You can see here it's like a maybe, but really you'll be able to identify what a molecule is based off of these 2 peaks alone. Okay, and in this case, it's ethanoic acid. Okay, so again, this is just straight memorization and application. There's not really much else to do here. Unless, yeah, you just have to memorize, and that's all you can really do in this situation. If you don't memorize it, your chances of getting a question, if this appears on the MCAT, your chances of getting it are pretty low, I think. But luckily, it doesn't appear, I think, too often, but I think it does consistently appear on the MCAT, but not very much. So you'll probably see like a question or two on every MCAT on something like this, like the IR spectroscopy, but probably no more than that. Next up is proton NMR, and this one is the exact opposite, where it's pretty much very little memorization, but it has a lot of actual like thinking and reasoning and like solving it out and writing it on paper. I think is very helpful for this type of problem. Okay, so proton NMR, as the name suggests, is you're measuring the hydrogens, the H pluses or protons, in a molecule, and you use that to kind of identify what the molecule is or what the structure is, roughly. So you can see here, this is color coded. These 3 blues apply to this. These 3 greens apply to this, and these 2 reds apply to this. Okay. Now, what are all these peaks, and what does all this mean? So there's 2 things that you have to look at for proton NMR, and only 2 things. Firstly, the number of neighbors, and secondly, the number of equivalent hydrogens or equivalent protons. And we'll cover that here, and I'll come back to this screen in a second for you guys to see, but I want to talk about this. So the n+1 rule, this is the neighbor rule. This rule states that the number of peaks in the NMR will correspond to the number of neighbors proton has, which is n, and 1 extra, so n+1. Remember, a neighboring proton refers only to protons on neighboring carbons, not protons on the same carbon. Okay, so let's see. Neighbors. We'll start with this blue one first, this hydrogen. How many neighbors does, and this corresponds to this peak, is color coded blue. So how many neighbors does this blue hydrogen have? Well, neighboring carbons. So it's on this carbon. So we're looking at this carbon. Now, how many neighboring carbons does this carbon have? Well, to the right, there's nothing because it's an end carbon, but it has to the left here. Okay, has this one right here. So this carbon has 2 protons on it. Okay, that means this blue hydrogen has 2 neighbors, has 2 neighboring protons, right? Because it has this one and this one. These, remember, don't count. These are on the same carbon. Has to be on neighboring carbons. Has 2 neighbors, therefore the n is 2, n+1 is 3. Therefore, this blue line should have 3 peaks. So 1, 2, and 3. 3 peaks, exactly. Okay, simple. Now let's talk about this one. Or sorry. Talk about this hydrogen, and I'll go through this one a lot quicker. How many neighbors does it have? This carbon has only 1 carbon neighbor. This one, because this is an oxygen. Oxygens have nothing, unless it's in alcohol, in which case it does count. Okay. Remember, alcoholic hydrogens also count. Carbons. This neighboring carbon has 3 on it. So that means this red hydrogen has 3 neighbors. n+1, 3 + 1, 4. So that means the red one should have 4 peaks. 1, 2, 3, and 4. 4 peaks, right? This one, oh, sorry, again, sorry. This green hydrogen, how many neighbors does this carbon have? Well, it has 1 neighbor. It's an N carbon, it has 1 neighbor, but this carbon has no hydrogens on it, no protons, it only has 2 oxygens. Therefore, this green hydrogen has 0 neighbors. So 0 + 1 is 1, so it's just 1 peak. Okay. Hopefully that clears up the neighbor rule. The n+1 rule. That's the first half. The second half is the size/intensity of the peaks. The size/intensity of the peaks is determined by the number of equivalent protons that peak represents. As a rule, all protons on the same carbon will always be equivalent to each other. But protons on different carbons may also be equivalent if the molecule is symmetric. And I'll give you an example of this. For example, if I had a benzene ring with an OH, well, let's say actually for simplicity, with a double bond O, okay? We had this molecule, and you can see that each of these has hydrogens on it. I'm not going to draw each one, but all of them have 2 hydrogens on them. Obviously, this hydrogen, and I'll actually do different colors. This hydrogen and this hydrogen are equivalent because they're on the same carbon, right? So they're exactly the same, right? It doesn't matter. They're basically the same hydrogen because they're in the same position. They get the same amount of spin and shielding and all of that, which is the principle behind NMR, but you don't really have to know that. The principle is like it provides a spin and it gets shielded by its neighbors, but again, not important for the MCAT. You just have to read the graphs and draw the molecules based on the graph. Okay, so these two are the same, right? Because they're basically in the same position. But this molecule down this line is symmetric. Therefore, these 2 hydrogens are also equivalent to these. So all 4 of these hydrogens are essentially the same hydrogen. They're in the exact same positions. They're equivalent, right? So there's actually 4 equivalent of red hydrogens. Now let's do blue. Obviously, again, these 2 are the same. These 2 hydrogens are the same because they're on the same carbon. But again, they're on symmetry. So these are also blue. And lastly, green, just these 2. And because they're on the point of symmetry, they don't have anything to be symmetrical with. So it's just okay. So the reds are equivalent, the blues are equivalent, the greens are equivalent. And that works because there's symmetry. If you had, for example, something like this, where it was just like a double bond O, and then you had a F here or something like that, right? Like a fluorine or something. And then you had carbon here. There is no longer a line of symmetry. So these are now different. So these 2 are still reds, but now these 2 are greens. And these are also not the same. So these are now blue and purple. Okay? Because again, no more symmetry. The symmetry has been broken. So the hydrogens here are now all different. Okay? So only when there's symmetry, carbons that are symmetrical can be equivalent. Now let's talk about what it looks like on the graph. So again, I mentioned the size and intensity. So it's color coded here, just like I did. So all of the blue hydrogens, there's 3 blue hydrogens that are all equivalent to each other. So that means these 3 peaks have to have high intensity, let's say, or the intensity equal to 3 hydrogens. These only have 2 hydrogens, so it has lower intensity. These 3 greens are again 3, so it will have higher intensity. And the intensity between these 2 should be equal, right? But you notice that this peak is big, this peak is smaller, but this has 1 peak, this has 3 peaks. Really, if you put these 3 peaks together on top of each other, it would equal this one big green peak, but yeah. Okay. Remember, these should be equivalent in terms of intensity. Okay. All right, moving on. And that's all it is, proton NMR. So there's 2 things. These 2 things you have to keep in mind: intensity with equivalent carbons, and number of peaks, which is the n+1. Okay. Remember, neighbors, neighboring carbons, and equivalent carbons. That's really all. Okay. Next, let's move on to carbon NMR. Carbon NMR is similar to proton NMR in that it helps identify the different carbons that exist in a certain compound, but there are some distinct differences. Main difference is that in carbon NMR, rather than the number of peaks, we look at the position of the peaks on the x-axis to determine what kind of carbon bond exists. Carbon NMR, if I had to describe it, it would be a combination of proton NMR and IR spectroscopy. That's the best way, in a sense, to describe it. And this is what it looks like. So depending on what kind of carbon bond you have, whether it's carbon double bond O, like a ketone, an ester, benzene ring, a double bond, a triple bond, a bond with a halogen, or just a regular single bond, has a different position on the x-axis in parts per million. Again, this is another chart that you have back to just memorize it, and that's it. It's another chart that you should be memorized, and be careful. There are not many patterns between this list and the previous one for the IR spectroscopy. Remember, this list also had some bonds, but there's not too many patterns between these. Maybe if you see some, go for it, but just be careful not to get mixed up between those two lists. These two lists are separate. They should be memorized separately. Okay. Another important difference between proton NMR and carbon NMR, that you are allowed to ignore the intensity in carbon NMR for the MCAT. It is not related to the number of equivalent carbons like it is in HNMR. So even if you have multiple equivalent carbons, they're not going to increase the intensity. That's not how it works in carbon NMR. It's based on something else. It's based on like protons, but it's way too complicated for the MCAT, and they don't ask that usually. Okay, so this is what it would look like. This is what a carbon NMR would actually look like, and it's labeled here. So C1 is here, C2 is here, and you can see that it's the ester, right? Then C3 and C4 are relatively close to each other. This one is this have an O bond, so it's a little bit more to the left, whereas this is just a carbon-carbon bond for 3, for 4, so it's just, yeah. Okay. Basically, again, just memorization and application. Carbon NMR, I think, is the least common one out of the HNMR and IR spectroscopy. It's the least common one, but sometimes it does appear. Another thing to note, you usually use C13, which is the radioactive form of carbon for carbon NMR, so just keep that in mind. If you see C13 NMR, that means just carbon NMR. Next up, let's talk about mass spectrometry. Mass spectrometry is a technique that separates molecules based on a mass to charge ratio, after which they can be ordered in relative abundance, as seen here. This can be used for isotopes, which is shown here, right? Remember, isotopes, elements exist in different masses. Since the atomic form has the same charge, you can essentially eliminate the charge ratio, so you can just order them based on mass because they all have the same charge. So the mass to charge ratio will just be essentially based only on mass. There's only one variable, okay, and then you can just order them based on mass. And then the relative abundance you can just measure out, right, once you have them separated. Or it can be used for mixtures of very similar molecules with uniform charge. Again, remember, unless you have a uniform charge, it's a little bit hard because there's two variables, so it's a little bit harder to separate into order, as in terms of a separation. But if they have a uniform charge and they're very similar, you can essentially separate them from a mixture. Okay, and that's more for biology than chemistry. The mixture and the isotopes is more obviously for chemistry. Okay, so that's about it for the lecture today. Like I said, this is a very short one, and a lot of these lectures I think will be relatively short. They're just filling in the blanks essentially for some of the lab techniques that we may have not covered. Okay. All right. So again, thank you very much for watching, and have a great day.